Good evening, everyone, and welcome to the Fralin Biomedical Research Institute's Maury Strauss Distinguished Public Lecture Series. Before we get started, and I have the pleasure of introducing this evening's speaker, I want to put in a plug for the upcoming talk, and I want to also thank Mr. Maury Strauss for his generous support of this program. Maury, it's a great pleasure to have your support. It means so much to so many people. Thank you. The next speaker will be April 15th. And it will be Dr. Christine Stabell-Ben, who's a professor at the Center for Global Health and chair of the Danish Institute for Advanced Study at the University of Southern Denmark. And Dr. Ben will speak on how vaccines train the immune system in ways that no one ever expected. Obviously, a timely topic for today, as is this evening's talk. So without any more delay, it is my great pleasure to introduce this evening's speaker, Dr. Michael Young from Rockefeller University, where he runs the lab of genetics. Dr. Young did his undergraduate work and then his PhD at the University of Texas in Austin, his undergraduate work with Burt Judd in biology, and then his graduate work in genetics as well. And then he went on to do his postdoc at Stanford in biochemistry department with David Hognes. And then after that, he joined the faculty at Rockefeller. He became a professor there, and along the way served for about a decade as a Howard Hughes investigator. He served as director of Rockefeller's National Science Foundation Science and Technology Center for Biological Timing, and he was named as the Richard and Jean Professor at Rockefeller, and he also serves as their vice president for academic affairs. Dr. Young has served nationally in many important leadership roles. I'm not going to name them all, but includes serving on the National Science Foundation Advisory Board for Genetic Biology and the Research Advisory Committee for the Center for Biological Timing, as associate editor of leading journals like Neuron and the Journal of Biological Rhythms, and on the New York Academy of Sciences Board of Governors. He's received numerous prizes and recognition for his scientific contributions, including the Hayashi Prize from the University of Tokyo, the Ashoff Pittendrigg Prize from the Society of Biological Rhythms, the Gruber Foundation Neuroscience Prize, and the Canadian Gairdner International Award and the Wiley Prize. He's been elected to the U.S. National Academy of Sciences, the American Philosophical Society, and he's a fellow of the American Academy of Microbiology. In 2017, Dr. Young was awarded the Nobel Prize in Physiology or Medicine, along with Michael Rosbash and Jeff Hall, for their discoveries of the molecular mechanisms controlling circadian rhythms. Using Drosophila or fruit flies as a model organism, they isolated genes that control normal daily biological rhythms and showed these genes encode proteins that accumulate in the cell during the night and then are degraded during the day. Subsequently, they identified protein components of the machinery, exposing the mechanisms governing the self-sustaining clockwork inside cells. As I look back over Dr. Young's CV, I mean, there's so many truly classic and important seminal papers. And just a couple I have to mention. 1984 is Nature and PNAS papers on the molecular substrates of biological clock in Drosophila, and then using gene transfer to restore circadian behavior rhythms. Their 1985 Nature paper studying the conservation of the unusual coding sequence from Drosophila clock gene, amongst other vertebrates as well. And then they've gone on to identify various mutations in this gene, including timeless and the effects of that on the clock and the behavior, including those that lead to shorter and longer period circadian rhythms. And then going on looking at nuclear localization signals, and then looking at mutations in human circadian clock gene, CRY1, in familial sleep phase disorders as well. So if I have to characterize Dr. Young's work, other than it's incredibly impactful, elegant, sophisticated, to me, it not only captures the essence of what biology is all about, that is curiosity and learning things about fundamental processes in molecular and cellular signaling, but then taking something all the way across the phylogenetic spectrum to understand a number of functions and dysfunctions together. It's the sort of thing that draws a lot of us into the life sciences, gets us excited about the fundamentals as well as the applications. So without further ado, it is my great pleasure to welcome Dr. Young. Please join me in welcoming him.
Thank you, Michael. I hope everyone can hear me okay. It's a great pleasure to be here. I decided today to use an unusual backdrop. What you see behind me is a is a set of chromosomes from Drosophila. And these are polytene chromosomes from certain cells in the larva of Drosophila that have been used for many decades to locate genes. And in fact, it's in large measure because of the availability of these chromosomes that we were able to isolate the genes that I'll be talking about today from Drosophila that regulate circadian rhythms. So let me share my screen. Okay. So let me just start by showing a record that's intrigued me for many, many years. These are data that were collected in 1960 by a colleague, Pat DeCourcy, who studied circadian rhythms in a variety of rodents. And what you're looking at is an example of running wheel behavior produced by a rodent that's in constant darkness in a laboratory setting where all environmental cues are being controlled. Constant darkness for 25 days, and each horizontal line is a day in the life of that animal. And the animal is given ad lib access to food and water, and has a running wheel, and makes tremendous use of that running wheel every day. And when or every night, I should say, since it's a nocturnal animal. Every night when it's on the running wheel, you see these deflections from this horizontal line. And the absence of those deflections mean the animal is quiescent, most likely sleeping. What fascinated me and still amazes me to this day is the regularity with which these running wheel activities are initiated and offset. So each day you can predict within a couple of minutes when the animal's gonna jump on that running wheel and start moving, and with about the same accuracy getting off of that running wheel. And again, this is in the complete absence of any environmental time cues that are available to the animal. Further evidence that the animal isn't picking up on something in the environment is the shape of these data. So every day, the animal is getting on this running wheel a little bit earlier than the day before, and getting off the running wheel a little bit earlier. So the period of this running wheel activity is not exactly 24 hours. It would be 24 hours if we introduced an environment of ordinary day-night cycling, 24-hour, 12 hours of light, 12 hours of darkness. You'd have a perfect 24-hour rhythm. But you take that away, and the animal displays what we can only imagine is an endogenous rhythm. What is the natural internal timing function that the animal has? It has a period that's shorter than 24 hours, which is, of course, why these are called circadian rhythms, from the Latin about a day, because that's what they are. They vary from species to species, never exactly 24 hours, but always aligned to 24 hours in a natural environment. Now, in the 1970s, as people began to pick up new tools to ask questions about the origin of this kind of behavior, the regulation of this kind of behavior, attention was turned to the brain. These are serial sections of a rat brain that were analyzed by Bill Schwartz when he was at the University of Massachusetts. And what Bill did was he gave the animals water that was that included radioactively labeled deoxyglucose, which is taken up by different areas of the anatomy of the brain and deposited according to the kinetics of glucose utilization. And what you see is there is a subtle change from day to night throughout the brain, But most particularly, there's an area in the ventral brain called the suprachiasmatic nucleus, part of the hypothalamus, which is very active in this metabolic activity during the day, but essentially inactive, or at least with regard to glucose utilization, essentially inactive during the night. So we have this oscillation in metabolic activity revealed by this experiment. Now, this led investigators to ask what the role of that part of the hypothalamus might be, and surgical experiments were performed in which the SCN was ablated and the behavioral rhythms of the animal were followed. That resulted in the finding that the SCN was necessary for the production of circadian locomotor rhythms. Remove that, ablate that area of the brain, the animal becomes arrhythmic. But perhaps even more striking, it was soon found that you could restore those rhythms by removing the SCN from a donor animal and transplanting them into into a hypothalamic ventricle in that ablated animal, and rhythms would be restored. The animal would not be able to follow a light-dark cycle. They'd lost connections that were responsible for light sensing, but they would display an endogenous rhythmicity that matched the period of the donor animal. So this was the beginning of a search for the origin of this kind of regulation. And of course, the next huge question was, well, what's going on in those tissues, in those cells, that's providing information about timing? Now, in the early 1970s, just as I was beginning graduate school, there was a striking study from Caltech by Ron Konopka and Seymour Benzer. Ron was a graduate student in Seymour's laboratory. Seymour was a very well-known molecular biologist, had done extremely impressive work in microbial genetics over many, many years, but had moved into questions about behavior and genetics of behavior, and had focused on Drosophila melanogaster in those studies. And what they published was a paper that reported the isolation of 3 mutants that no longer had the normal 24-hour or nearly 24-hour rhythms, circadian rhythms of a wild-type fly. One mutant was arrhythmic, another had a period of 19 hours, so it was a short period, a fast-running clock, and the other had a period of 28 hours, so a long period mutant, presumably a slow-running clock. Another impressive finding was that these mutations affected both the locomotor activity of the fly and also some developmental changes in the fly that were known to follow a circadian pattern. So this regulation had to affect more than one organ system in the animal. Probably the most important feature of their studies was that they had found that all of these mutations seemed to affect the same functional gene on the X chromosome, and that came from standard genetic complementation tests that that you'll understand as I go on. So as I was saying, this was work that was done just as I was entering graduate school, and lucky me, I happened to be studying these chromosomes that I mentioned before. And in fact, I'd been studying a region of the X chromosome shown in this box that is shown here. The chromosomes of the fly in these particular cells are so well mapped that individual bands have number designations, and these are consistent patterns from animal to animal, and even from tissue to tissue within the same animal. And saturation mutagenesis studies had indicated, had placed about 150 mutations in a region containing about 20 of these visible DNA-rich bands in these chromosomes. And we'd been studying those, trying to understand the relationship between DNA and genes in this simple eukaryote. The mutations that Ron and Seymour had identified fell roughly in this region, but I realized that Drosophila had a system, these chromosomes, that might allow me to precisely locate these new mutations that seem to affect a functional behavioral gene. And the next slide shows a result of an analysis that I did as a student using chromosome rearrangements. These were breaks that would separate different intervals, subintervals of this region. Again, this is the interval that I pointed out in that box in the prior slide. And what you'll notice is that we've got these dashed lines that connect chromosome breaks to different regions of this chromosome. And I was particularly interested in breaks that were occurring here. And this collection is meant to emphasize breaks in that area, because all of these were mutations that affected Ron and Seymour's PER locus. One of these, four of these mutations were deletions that ended adjacent to the period locus. One of these mutations was a reciprocal translocation, so it simply broke the gene in half. It was a PER mutation. None of these mutations at the time that they were originally studied was appreciated to affect a behavioral gene. And in fact, when I tested all of these mutations, I found not only behavioral effects, but I also found that all of the other mutations that had previously been mapped into this region, none of them had affected behavioral rhythms. They didn't fall in this particular gene, which also indicated that this gene's only phenotype that had been defined so far were associated with circadian behavior, the original 3 mutations plus all of these chromosome rearrangements. Now, the most valuable part of this early study that I wanted to point out is that Ron's mutations, and for that matter, most mutations in any organism being studied in those days were point mutations. Certainly Ron's and Seymour's mutations were point mutations. And in the early 1980s, late 1970s, early 1980s, there was no way that you were going to clone a piece of DNA and walk for tens of thousands of base pairs looking for a single base pair change to identify the location of the gene. So these were incredibly important in identifying exactly where this gene was positioned within this chromosomal region. And in fact, we and our colleagues at Brandeis, Michael Rosbash and Jeff Hall, used this information about chromosome rearrangements to precisely identify the period locus, the first gene to be identified affecting behavior. And in addition to realizing that period corresponded to a single transcription unit that made a detectable single poly-A messenger RNA, we found that we could take that single gene and prove that that was the gene responsible for those period phenotypes by transplanting them into an arrhythmic animal and asking what's the impact on behavior. And sure enough, what we found and what Michael and Jeff found was that it was sufficient to transfer that one transcription unit into an arrhythmic animal and restore rhythmicity, indicating that that must be the gene and that there was nothing especially unusual about behavior, that you couldn't transplant a gene that controls behavior just like you would transplant a gene controlling eye color. So this excited us. In addition, the finding, as I said before, that this gene seemed to be unique in the region, that there were no alleles, there were no versions of this gene that affected anything other than circadian rhythms, the fact that the mutations that we had available to us could stop clocks or make clocks run fast or slow, all sounded like this might be a gear in some kind of a clock mechanism. But how do we go forward from there? So our approach at that point was to say, well, let's see if we can find other genes in the system that might help us piece together a mechanism that might tell us more about how this behavior is controlled. Now, our early efforts in this were not particularly successful. Ron, as a graduate student, had found his first period locus mutation in the first 200 fly strains that he sampled. This is a Photoshop rendition recreating an experiment that Amita Segal and Jeff Price did in our lab, where they had to go through 7,000 strains of flies, mutagenized flies, to look for, to find a single new mutation affecting circadian rhythmicity. To understand the magnitude of this experiment, I should point out that each one of these bottles contains a different strain of flies, but in order to find out if rhythmicity is affected, you have to sample that bottle several times a day for multiple days. And so you can imagine that 7,000 quickly gets multiplied into a lot of Drosophila handling. Nevertheless, it all proved very worthwhile. We found a second mutation. It was a new gene. It was on the second chromosome instead of the X chromosome. We named the gene timeless. It was an arrhythmic mutation that resembled in many ways the original period mutation. And this second gene became of tremendous interest to us because we realized it had genetic and physical interactions with the first gene, period. As I'll explain in a minute, one of the things that we eventually found is that the two proteins made by period and timeless come together as an essential part of the clock mechanism in the fly. So we had not entered this genetic screen looking for things that would alter the effects of permutations, for example. We weren't doing a modification screen. We were looking for anything that would affect circadian rhythmicity. So we were struck that the very next thing that we isolated brought us back to the same system that included the original gene, period. So now we have two genes working in the same system, period and timeless. Now, by continuing that kind of an effort and using some new techniques to measure locomotor behavior as opposed to the hatching assay that I just spoke about, we were able to come up with another 10 or 12 genes that were critical to the operation of circadian rhythmicity. And the most important feature of all of that, the results of that screen, which took place in our laboratory and also in the laboratories of Jeff Hull and Michael Rosbash, is that everything that we discovered fit back into the original cluster of period and timeless. So we only could define a single mechanism that was involved in circadian rhythmicity in the fly. Everything that we identified by genetic screening, and again, all of the genetic screening was just give us mutations that affect circadian rhythms without a bias. They were unbiased screens, and yet they all came back to the same system. So this is I'll give you a skeletal overview of how the system works. We have two transcription factors, clock as C and cycle as B, which form a complex that interacts with promoter regions on the period gene and the timeless gene, initiates transcription from both of these genes simultaneously, same time of day. Those transcripts are translated to give period and timeless proteins. If those two proteins can get together and form a period timeless biochemical complex, then they take on a new property of movement to the nucleus. They're able to move to the nucleus as a paired product. And in the nucleus, they interact physically with cycle and clock, inhibiting the action of those transcription factors to shut off their own genes. Now, cycling in this system comes from other activities that occur in the cytoplasm after these transcripts are made. So the first break I'll talk about is a break on the accumulation of the period protein. As period proteins are translated early in the cycle, when low levels of timeless protein are available, most of the partnerships being made are made with a kinase that we found called double time. This is an ortholog of casein kinase 1 in mammals. And double time physically associates with period, phosphorylates period, and that phosphorylation on certain sites leads to ubiquitination and proteasomal-based degradation of period protein. That pattern of modification and degradation is circumvented if period time pairs with timeless. So a per-tem complex is immune to those modifications that produce period degradation, allowing this nuclear transport. On the other hand, we also have a break on the accumulation of timeless. And that break is provided by a novel photoreceptor called cryptochrome, which is related to bacterial photolyases. It's a light-absorbing protein. In the presence of daylight, or light of a wavelength comparable to daylight, cryptochrome conformation is altered in such a way that it now interacts with the timeless protein. Both proteins become phosphorylated, ubiquitinated, and turned over by proteasomal degradation. So when the lights are on, we can't generate timeless. We can have loads of timeless RNA, but we won't generate stable timeless proteins. And of course, in the absence of timeless proteins, all the pairing of period proteins is going to be with double time. So all day long, we have transcription, we have RNA being produced, but we have no period and timeless proteins being accumulated. We can't come back and stop this process from continuing. At nightfall, cryptochrome is now inactivated, timeless can accumulate, and timeless will eventually start rescuing period proteins, produce these per-tem complexes that move to nuclei and can now bring us back to the beginning of the cycle where clock and cycle are inhibited and there's no transcription on period and timeless. And of course, as those proteins turn over, because they're not being replenished by new cytoplasmic production of period and timeless, will reinitiate this cycle. Now, I've already told you about two features, the double time effects on period and cryptochrome's effects on timeless that produce delays in the system, cycles in the system, and certainly the double time delay or effects will be present even in constant darkness to give oscillations even in constant darkness. So we see mechanisms emerging that tell us how this system is entrained to light-dark cycles, but also how this system oscillates in constant darkness to give its own intrinsic periodicity that's set by the nature of the species. Now, if this mechanism were just spinning on its own, we would not really understand much about how circadian behavioral rhythms or physiological rhythms are produced by these cycling mechanisms that involve just a handful of genes in their proteins. But we do, if we look at the transcriptome of Drosophila, if, for example, we examine the Drosophila head and say, are there cycling genes other than those that I've just talked about? In the Drosophila head, we see hundreds of these genes that are cycling in a rhythmic, in a circadian fashion. What this are the results of microarrays that we conducted about 20 years ago. With 150 genes are being analyzed here. Each gene is represented, its pattern of expression over 6 days is represented in a horizontal row with high levels in red and low levels in green. And the peaks each gene cycling determine where on this matrix it's placed. So genes that are produced at highest levels around dawn are near the top. Those peaking at around noon are in this region. Those at dusk down here, midnight down here, and so forth. So what we have here is a panel of closest neighbors with regard to the phases of oscillation. But again, we've got this enormous body of genes that are oscillating in the fly's head. And these cover every imaginable area of the animal's physiology. Behavior is just one component of the regulation that we see when we look at the transcriptome. The other thing I want to point out is that if we look at the waves of transcription that go top to bottom here, the red and green stripes, you notice that those are going diagonally across this map. What that's telling us is that these rhythms are not simply swinging between a nighttime feature and a daytime feature. It's much more like a music box where there's a continuous changing of notes across the day and night, so that at every hour of the day, there's a unique combination of genes that are hitting their high and low points and presumably defining certain states of physiology in their respective cells. Now, this entire pattern is dependent on those few clock genes that I just previously mentioned, and that is shown in this pair of slides. This is a reproduction of what you just saw. So this is in a wild type animal. What we get in a 6-day experiments, we get 6 stripes of rhythmicity of these 150 genes. If we look at the same genes for 3 days, now this is compressed, so it's a little tighter, but for 3 days, we're looking only during light-dark cycles at the patterns that are produced in 3 mutants: timeless, period, and clock. So these are all arrhythmic mutants behaviorally, but what you see is at the molecular level, they are arrhythmic as well. We don't see any rhythms in the molecular features of their clocks in the components that I spoke about previously, but we also have lost rhythmicity for all of these other genes. Nothing that we see at the transcriptional level is oscillating anymore in these 3 mutants. So this is confirmation that there's a very robust effect of these few genes on the large pattern of transcription. The circadian clock is essentially orchestrating the rollout of gene expression in many different cells and of many different genes to give this hour by hour changing arrangement of gene activities. And the last thing I want to point out is that these experiments are conducted, the original experiments are conducted both in constant darkness and in light-dark cycles. These are only light-dark cycles. Here we saw that the rhythms occurred whether or not the light cycle was going or not. Here we've got the light cycle going, and yet we see no rhythmicity. Now, surely these rhythms arose as an adaptation to the rhythms in our environment, in Earth's environment. But what this kind of an experiment tells us that after a billion or so years of generating life on Earth and producing mechanisms such as circadian clocks, information from light-dark cycles is now filtered through this molecular mechanism that is now essential for the representation of the outside world, the oscillations in the outside world, into transcriptional rhythms. Transcriptional rhythms can't occur directly in response to changing environmental lighting. Now, another thing that I want to point out briefly about seeing all this oscillating gene activity downstream of this central clock mechanism that's of interest medically is that there are huge numbers of genes, just as in Drosophila, huge numbers of genes that are being expressed with the circadian rhythm in mammals. So these are data that were produced by John Hoganish at Cleveland Medical Center a few years ago. And what John shows us is that of the 100 best-selling drugs in the United States, more than half, 56 of them, have rhythmic gene targets. Now, when you look at the number of rhythmic genes in the animal, that's not surprising because of all the genes in a mammal, more than half are rhythmic, and therefore we shouldn't be surprised if many of our drugs are hitting rhythmic targets. More importantly, though, many of the drugs that are being used on these targets have very short half-lives. And in fact, almost half of the genes that are hitting rhythmic targets themselves have half-lives less than 6 hours. And this is a list from that group of 100 best-selling drugs that have half-lives of less than 6 hours. So imagine, if you will, a pill that you take at night before bedtime at 9 or 10 o'clock at night. But imagine that you're trying to, the target of that drug is a gene that has its peak at 6 p.m. or 7 p.m. It's almost 24 hours before you come back and hit the peak with that drug. This has to severely diminish the effectiveness of such a drug. You begin to wonder what dosing would be all about. So this is, I know many of you already appreciate the importance of better understanding the relationship between these short-lived drugs and their targets. So I want to turn now to some more recent work that we've been conducting that is now switching us from looking at Drosophila to taking advantage of what we've learned in the fly to learn something about human biology, human circadian rhythms, and human sleep problems, human sleep disorders. And one of the things I should preface this with is that once the fly genes were isolated, it became clear that orthologs could be found throughout the animal kingdom, certainly in mammals, certainly in humans. We find a closely related set of genes performing functions that are very similar to those that we originally found in Drosophila. So this is a very old system that goes back to the origins of the animal kingdom. So delayed sleep phase disorder, here you've got a sort of a cartoon depiction of what we're talking about. These are individuals that have a tremendously difficult time getting to sleep each evening. Here you've got someone maybe cramming for an exam late at night the day before an exam, but we're talking about lifelong assaults on the wake-sleep cycle. And in fact, the syndrome, delayed sleep phase disorders, is known to affect probably around 5% of the population in the US. It's a persistent delay in the timing of the major sleep episode and has shown great resistance to attempts to advance sleep phase. So because of its abundance, its prevalence in many populations, including the US, we wanted to learn more about this and whether there might be connections to any of the genes that we know the most about. So let me first show you some activity data that will better describe the syndrome that we see. So we have two records, home actigraphy and sleep log records. On the top is a control individual, and in B is a DSPD individual that I'll be telling you much more about. And just like those rodent records, we've got, in this case, an activity watch on the wrist of our subjects. And we also ask them to keep a sleep log and tell us when they get in bed each evening and when they get out of bed in the morning. So our control individual, you can see, is very active during the day, is relatively inactive at night, even though there's going to be some movements of the wrist while you're sleeping. But a very consistent record, log record for getting into bed and getting out of bed. Out of bed is typically 8 or 9, 7, 8 or 9 in the morning, and in bed's 11, 11:30 every night. Now, if you compare that record to the record that you see down below for a DSPD individual, you see what many of you may see chaos. There's activity at essentially all times of day if you go far enough into this record. And if we look at the sleep log to help us interpret the activity record, we see that this individual is getting into bed well after midnight every night. In some cases, getting into bed after dawn, but certainly deep into the night compared to our control individual. Out of bed times approach noon, 10 o'clock, 11 o'clock in the morning are typical for this individual. So quite different pattern from what we see in the control subject. So we came across this individual as part of a collaboration with a group at Weill Cornell, Scott Campbell's group at Weill Cornell, and we began some molecular studies and further genetic studies in this individual's family. A number of the individuals in the family were interested in the work we were doing because they also were complaining about sleep problems. So this is the proband. This is the individual whose record you just saw. And these are relatives of that individual, a cousin over here. All of those that have a darkened circular square are females or males that show a sleep disorder clinically. And the individual that is non-colored does not show a sleep defect. What we did at this point was whole exome sequencing to look at the structure of every protein in each of these individuals to see if there might be a protein associated change in any of them, or that would give us a clue about what might be responsible for this pattern. It was clearly behaving as if it were an autosomal dominant mutation that was being carried in the affected individuals. And what we found was a hit in a gene that was quite familiar to us, cryptochrome 1. And that cryptochrome 1 mutation, the same mutation, was found in all of the affected individuals in that family. The mutation is a single nucleotide change, and it occurs just downstream of one of the protein coding regions, one of the exons of cryptochrome 1. And in fact, the nature of the change suggests that the splice donor region of that for exon 11 has been damaged so severely that exon 11 can no longer splice to exon 12. We're not going to be able to make a full-length protein anymore. That individual should have difficulty making a full-length protein. So it in fact predicts that exon 12 will be joined to exon 10 in making messenger RNA, splicing messenger RNA. And in fact, if we look at a number of individuals, some of which are DSPD carriers from other families, some are controls, but we see only one. Individual 11, who's the individual I've been talking about, shows two versions of this RNA and two versions of this protein. And the sizes of those different proteins fit with our expectation that they carry two alleles of this gene, one of which is affected by this mutation and will make the truncated protein and the truncated messenger RNA. So at the molecular level, there is clearly an impact of this mutation on the structure of the protein, the cryptochrome protein being made in all of the affected individuals. Now, we could determine whether or not that altered gene was in fact capable of producing an altered rhythm by moving an altered version of that gene into cell lines that could express a rhythm, but could only express a rhythm if there was a restoration of cryptochrome 1 function. So these are cells that do not carry any cryptochrome. We can either replace a wild type version of cryptochrome or this mutant version of cryptochrome to see if there is a measurable effect on the rhythm, on the period of the rhythm of those cells. And remember, these clocks are active in every cell in the body. And so we're able to monitor those rhythms physiologically as well as in whole organism behavior. What we saw is that in those cells that receive the truncated version, the mutant version of that gene, they have a longer period to their rhythm than those that receive the wild type version. They have about a half an hour to 45-minute change in the period of the rhythms that they're producing. So I need to tell you a little bit more about human clocks. They're largely the same as Drosophila clocks, but there is one important difference, and that is in humans and all mammals, so far as we know, the partner protein for period is no longer timeless, it's cryptochrome, which we remember was a regulator of timeless in Drosophila. So now cryptochrome proteins pair with period proteins, move into nuclei and inhibit the activity of the orthologs of clock and cycle, which in mammals are simply called clock and BMAL. So the other features of this cycling mechanism are much the same as in Drosophila, but we now have cryptochrome per complexes that are the inhibitory complex in this cycling mechanism. So because we now have cryptochrome interacting with period and cry-per complexes interacting with clock and BMAL, we wondered whether this altered form of cryptochrome would upset any of these physical associations that are so critical to the operation of this clock. So these are immunoprecipitation experiments that I'll just describe very briefly. If we start down here, we're doing immunoprecipitations of a hemagglutinin tagged clock protein in the presence of BMAL, its partner, and also in the presence of a tagged version, a MIC-tagged version of cryptochrome 1. But we have two different forms of cryptochrome 1 that we want to follow. We want to follow the full-length form. These are the inputs, the full-length form of cryptochrome, the truncated form, our mutant form of cryptochrome, and we want to make a synthetic heterozygote where we put into this immunoprecipitation mix in HEK293 cells. These are all cell-based assays. We put both full-length and the truncated mutant form. And we say, what do we immunoprecipitate now if we pull down clock? What comes down with clock? Well, what we find is that there's a great loss in the full-length protein coming down. There's a tremendous recovery of the truncated form of the protein. And if we look at the synthetic heterozygote, we see there's a strong bias for pulling down the truncated form of the protein versus the full-length version of the protein. We see essentially the same thing if we look at the input and the pull down where we've reversed this and we're pulling down BMAL in the presence of clock. So once again, we see a diminution of the full-length form either by itself or in combination with the truncated form. On the other hand, if we look at PER, the input of which is shown here, and the pull downs of which are shown here, if we look at what PER pulls down both forms of the protein equally. So it looks like period and cry proteins have no trouble coming together, associating with one another, but the truncated version of the protein has a greater avidity for its target proteins, clock and BMAL, which it will be inhibiting. So I want to tell you a little bit about some very recent work that Carrie Parch at UC Santa Cruz has done. She's done structural studies of cryptochrome, biochemical studies, looking at interactions of different parts of cryptochrome with itself, and has shown that the C-terminal tail, which includes exon 11 encoded protein, folds around the cryptochrome protein, and ordinarily exon 11 falls in a region on the surface of cryptochrome that is a docking site for the clock protein. So in order for cryptochrome to inhibit clock, it must dock with clock using this area that's shown in green. And ordinarily in wild type cryptochrome, the C-terminal tail folds around so that exon 11 is juxtaposed next to that clock interacting site. So apparently what we've done by eliminating exon 11 is we have taken an autoregulatory part of this protein away, increasing its activity as a transcriptional inhibitor, which fits with everything we've seen at the molecular level. I don't have time to go into transcription assays, but all of our transcription assays also indicate that it's an enhanced inhibitor of clock. So another striking finding, unexpected finding that we had looking at this mutation was its prevalence in human populations. So we began to look at databases from around the world. This is a database that's managed at the Broad. There are actually about 140 whole exome sequences, 140,000 as opposed to about 60,000 exomes that would have been sequenced to give the data shown here. But the information that's shown here has not changed very much. Importantly, this is not a tremendously rare mutation. It's present at about 0.5%. About 0.5% of the alleles, cryptochrome 1 alleles, carry this mutation. Now, since this is a dominant mutation, the carrier frequency is going to be about twice that. So about 1% of the world's population, if we put everyone together, would be the frequency of this of carriers. Perhaps even more interestingly, if we look at Europeans, non-Finnish Europeans, we find that about 1 in 75 individuals will be a carrier that includes this mutation. So this raised the question to us about whether or not what we found in this original family would be seen if we identified additional carriers in other populations from elsewhere in the world. We formed a collaboration with a group in Turkey, at Bilkent, Turkey, Tayfun Öztürk's laboratory. And he had access to a large database of individuals and identified 6 or 7 families pretty quickly that had carriers of this mutant form of cryptochrome. And what we were able to do using those families is to conduct sleep assays on all members or most members of these families and sort them into those phenotypically that appeared to have a sleep disorder versus those that didn't, and then genotype them to determine whether there was a relationship between those with a sleep disorder and those that carried this altered form of cryptochrome as we'd seen in that US family. And I think everything is summarized down here in the bottom right corner. If we just look at the mid-sleep time for the 2 sets of individuals that come collectively from all of these families, we find that those that don't carry the mutation typically have a mid-sleep time of about 4:30 in the morning, whereas the larger, the most common area for mid-sleep time is about 6:30 in those that were carriers. Although I should point out that there was a much wider distribution of sleep patterns in the carriers than there were in the controls, and this is something we're interested in. This is not just simply delayed sleep is an incomplete definition of the phenotype that we're seeing here. So let me summarize by saying that we've looked now at a number of unrelated families and the presence of this mutation, which we refer to as Cry Delta 11, faithfully predicted DSPD. It seems to be a rather high penetrance for this allele. And so while it certainly isn't going to account for all of the DSPD that's been seen clinically, they're a small fraction of it should be broadly based on this version of the genetics worldwide. Since the mutation is found in multiple members of each family, different generations of each family, it certainly must be carried in all the tissues of these individuals. And so we're very interested in what the effects of these mutations might be on things other than behavior. As I pointed out, there's enhanced binding to clock and BMAL in mice and in human cultured cells. The delta 11 version of the protein appears to be an enhanced, strengthened transcriptional inhibitor. Now we understand that the tail is involved as an autoregulatory device. And I'll just point out that we're interested in clinical features of this mutation and the underlying genetics, but of a clinical problem. But interestingly, we've learned something very basic about the way cryptochromes work from looking at this human mutation. This is a new discovery about the way the cryptochrome tail works that has not been revealed by work in animal models. The competitive binding of clock and BMAL, or to clock and BMAL by this version of cryptochrome, suggests why this is a dominant mutation. It tends to elbow out the wild type version of the protein. Presumably we have a dynamic interaction between inhibitor and inhibited, and the on time for the variant is almost certainly higher than that of the non-variant, the wild type protein. And then finally, as I've shown, the variant expression of the variant is sufficient to change the circadian rhythmicity of isolated cultured cells. So we have very firm conclusion about the molecular impact of this mutation. So just in closing, I'll just say that, the sequence databases are up to about 140,000 whole exomes now. When we think about the coverage, you've got polymorphisms every 1 to 3 nucleotides in every protein imaginable in the genome. So if there are mutations that have an impact on sleep, a significant impact, a prevalent impact on sleep, it ought to be already present in these databases. So we're presently taking the tack of looking at candidate polymorphisms that are both prevalent and predicted to have an impact from the nature of the change we see, and so that we can address large numbers of subjects and confidently determine the human phenotype of such mutations. And of course, as we've done in this case, we feel we can look at molecular mechanisms even in cell-based models. We can either do this in animal models or in cell-based models. And then finally, a longstanding question has been, what's the relationship between sleep disturbances and the etiology of many clinically important human disorders, psychiatric disorders, metabolic disorders? If you're dealing with higher frequency mutations, there is the new opportunity to make connections between these, or to test connections between these with a high degree of confidence and reliability based on the numbers of subjects that you're able to analyze. So I'll just close by pointing out that all the molecular work on this last chapter of circadian mutation in humans has been done by a postdoc in the lab, Alina Potke. She's headed for Caltech. She's going to be an assistant professor at Caltech at the end of the summer. That work was a collaboration with Scott Campbell and Patricia Murphy at Weill Cornell Medical School. Anna Krieger helped analyze much of the data, also at Cornell Medical School. Tayfun Osalek and his student, Anur Anat, collected data for us from the Turkish families that we were able to analyze. And of course, this is the support we've seen. And finally, I just want to point out that we feel we've learned a lot about human and mammalian rhythms now, but I don't think we could have done that without our starting point, which is Drosophila and those chromosomes that you see behind me. So thank you very much for your attention.
Thank you very much, Dr. Young, for that outstanding presentation. We have several questions. I hope you'll be willing to take a few questions. So we had at least 2 or 3 questions from separate individuals about CRY1, whether that's a targetable place you can help people with DSPD, and how are people with this syndrome treated right now?
Right. So right now they're not treated. There's been variable evidence that strong environmental exposure may improve patterns of sleep. So, for example, we have a couple of construction workers that showed up in our database in Turkey, and they seem to be the least affected, clearly carriers, but they seem the least affected by these mutations. But I think we need a much larger database to find out. Certainly, there were enough individuals involved that had good exposure to environmental light cycles that's no panacea. Drugging, I think, is a great possibility, because you have 2 different versions of a protein, and I think we've all been used to hearing about drugs that can distinguish between 2 versions of a protein. Here we've got a protein that really, a variant protein that differs quite significantly from the parent protein. You're missing 24 amino acids, an entire exon's worth of amino acids. So a drug that would have an impact on that variant form versus the wild type form could be very useful. But even in the absence of that, it could be that any drug that would suppress the action of cryptochrome would have a beneficial effect, because we have a hyperactive protein that we're trying to that we would have to control. And we can imagine that the level of specificity might not have to be too high. If we can suppress a large fraction of the activity that's being produced, most of the activity that we're suppressing is going to be in those heterozygotes on that variant form of the protein. So I think none of these things have been tested so far. The right place to do it would be in cultured cells as a starter, and then in animal models. But I think we do have reasonable approaches to something like this. Also tells us how important it is, if you've got a behavioral disorder, to know the specific basis for the behavioral disorder. I suspect that kind of a treatment for this form of DSPD, while it may be impactful, would have no relevance at all to other forms of DSPD. So knowing the specific defect and the notions of personalized medicine, I think, are going to be prominent in any thoughts about this.
Thank you. And actually, as a bit of a follow-up to that question, one of our circadian rhythm researcher faculty here, Carla Finkelstein, has a question about. She says, "Why do you think chronotherapeutics are not more broadly applied in the medical field?" Yeah,
I'm familiar with her work. I know she's interested in these questions as well. And she and we are trying. There's I know that UC San Diego has a large group that meets often with representatives from pharmaceutical arena. And I think it's just the information trickles out or trickles in, is swept up slowly. And, it's not just about treatments, it's also about clinical trials. If you're doing a clinical trial, you need to know if something fails, did it fail because of not because of the dosing per se, but because of time of day. And, many of these clinical trials are conducted in rodents that are nocturnal. You've got to compensate for the fact that if you're taking a drug from a rodent animal model, then time of day could be critical to understanding success or failure. So I think it's not just patient treatment, but also clinical trials. And those are the easiest to think about controlling. You don't you can ask the question, does it matter what time of day we give the drug in a clinical trial? And that's not the same thing as writing out a prescription with a time of day that's generated by a lone doctor in their office trying to do the best they can to take in new information.
Great, thank you. Another one of our senior faculty members, Dr. Rob Gordy, yes, he says cryptochromes are also linked to magnetosensitivity in birds and flies. So does this hint at a deeper relationship between circadian rhythms and navigation? The human cryptochrome mutants have a higher propensity to get lost.
Yeah, no, that's a great question. I don't have a good answer. I do know that, cryptochromes there have been some migration studies for cryptochromes in migrating insects, butterflies, for example, but the results have been are not crystal clear. The one thing we do know is that many insects use their circadian clocks to navigate. And of course, cryptochrome will be in that mix, but what we know about the way those navigational signals are coming, we know this from very old work going all the way back to before von Frisch, for example, was doing experiments of this sort, showing that many insects, honeybees, get their cues from the location of the sun, but have to compensate for movements of the sun as they provide information to the rest of their hive mates about locations of food. So we know that circadian clocks are involved. What hasn't been picked apart is how individual proteins in these clocks might also have independent effects on wayward animals.
Thank you. One of our neuroscience faculty members, Reed Montague, said the clock mechanisms you've discovered and pursued relate to rhythmic patterns across relatively long time periods, hours, days. But do these same signaling systems or some congener of them regulate much faster timing mechanisms within and across cells? For example, it's important to learn all sorts of fast temporal relationships in order to seek food, escape danger. What is known about timing that must exist to underwrite such learning abilities?
Yeah, there is evidence that that was collected, but it's been argued in both ways that some of these proteins might be involved in high frequency rhythms. The biggest difficulty in imagining circadian clocks in some way wired into high frequency rhythms is that some of the principal components aren't there during the daytime. And that's going to be true in an insect in every cell because the insect is translucent. Cryptochrome is going to wipe out timeless proteins, loss of timeless, loss of per. So any involvement of these proteins in higher frequency rhythms would have to be through quite different mechanisms, I suspect. And I think it would be serendipitous if some of the same proteins that are involved in 24-hour rhythmicity or near 24-hour rhythmicity were involved in higher frequency rhythms. Certainly they're involved in longer period rhythms, such as photoperiodic rhythms. And those we can understand as using circadian clocks to help measure day length. But high frequency rhythms are still, it's a great area for students to jump into. I think if I were a student today, it would be on one of the things on my list to think about.
Thank you. I think we'll just, if it's okay with you, we'll do two more questions and we'll wrap it up. We have a lot of questions, can't get through all of them. One of them from one of our developmental neuroscience faculty, Anthony Lamontia, says, due to the transcriptional regulation of the pathway, how many mutations, either in Drosophila or humans, have been identified in upstream regulatory or intronic sequences of any of the individual genes?
Yeah, this for me was fascinating. When we did our random unbiased genetic screening, we were using chemical mutagens. EMS, for example, was one of the key mutagens that we used. And so we're trying to make point mutations. Of course, Konopka's mutants were also point mutations. So we've really hit the genome pretty hard. Our group and the groups of Jeff and Michael hit the genome pretty hard with this strategy of chemical mutagenesis that should produce a range of, many point mutations should not really be biased for hitting coding versus non-coding DNA. And yet everything that we came up with that had an impact was a protein changing mutation. Now that could of course be because you need something bigger than a point mutation to cause a regulatory change. So we'd have to go back with other methods to see if we could get something to happen and see if we get new information, new genes, new targets with that kind of a screen. But nevertheless, the statement holds that everything that we did find with by using mutagens that principally are going to produce point mutations were protein altering changes.
Okay, and then there was, I feel like I should pick this one. There have been 3 questions about this topic. So this from an anonymous person. It says, have you done genomic sequencing in individuals with cited non-24, like you've done with DSPD individuals? Do you think that the same mutation in cryptochrome could cause non-24 in humans?
No, we haven't done anything like that. I guess yeah, our approach so far has been not to pick another disorder. This, as I pointed out, we clearly started with DSPD and a group of DSPD patients initially. But since we were so successful in running to a genetic variation by going that route, we sort of, since the databases have gotten so rich, since they're so large, we've sort of turned things around a bit and are claiming a non-bias with regard to existing disorders and simply look at genetics first. Do we have variation that looks significant? Does it affect genes that we know something about? And collect things in that arena, which I think we have the molecular tools to analyze much more effectively. But certainly if non-24 were caused by the same mutation, I suspect we would have seen that given the numbers of subjects we've had a chance to analyze so far, particularly in the Turkish population. I mean, the numbers that we've gotten up to now are something close to 100, I think. And we're not looking for DSPD. We're just looking for subjects that carry that mutation. And then we analyze the behavior patterns that we see, and we haven't seen anything that looks like non-24. So if it's responsible for non-24, it's a much less frequent manifestation of a response to that allele.
Okay, thank you. And I guess I'll just wrap up by saying there are a whole bunch of comments in here saying thank you for a great talk. And I want to join with them in saying, Dr. Young, thank you very much. We really enjoyed the presentation.
Oh, thanks.
Michael, that's terrific. I will see you in about 15 or 20 minutes at a little online dinner, I think, right?
Okay, sounds